Hi, Alicia. Can you hear me?
Tim, we match. We're like twins.
That's funny. Nick, what happened? Where's your checks?
Oh, I can't hear. Wait, what did you say? I can't hear anything.
I said, Nick, what happened? You didn't get the memo. Where's your checkered shirt?
Yeah, no, I didn't.
Come on, Nick.
I have half checkers.
Same page here.
Yeah. All right, we have 28 people in right now. Do you know how many signed up?
It was 52 as of yesterday.
Yes.
Okay.
Yeah.
We'll give them a minute because we started it late. That's my fault.
All right.
I'll be right back. I'm gonna grab some water.
Okay.
Let me. I'm sharing my screen. Where, how did I do this last time?
Oh, on the share screen?
Yeah.
So when you click share screen, it's going to show you all the things on that you can share. If you look at the bottom left of that panel, there's an option, a box to check for share computer sound.
Yeah, but where do I click on the share screen? Why don't I see it up anywhere right now?
It could be at the bottom. Mine is at the bottom. Wherever your toolbar is with like participants, Q&A, polls, chat,
Right.
your screen is one of those.
Oh, here we go. Okay, sorry.
Okay.
It says that the host disabled participant screen sharing.
Okay, let me find you then. If I change the host to you, it's going to allow you to share. It might take me off of video, but if so, I'll figure it out and sign back in.
Okay.
Nope, it allowed me to stay. So now you'll be able to share.
Okay. You guys got that?
Yeah, it's perfect.
Got it.
Looks great. All right, we're up to 40 and it's just after 1, so we can probably get started if you guys are ready.
Yep, ready.
Oh, okay, I will. All right, ladies and gentlemen, thank you for coming again to another CLE. Today we're going to have Alicia, Lilley, and Tim Crino again. They're going to be talking about DNA Investigation Basics. Again, it's part of our newly established forensic unit, and I'm looking forward to it. I think we can all use, these are really complicated concepts. We can all use a lot of instruction on this, so I'm excited for it. And I think with that, I'm just going to turn it over to Tim and Alicia and let them get started. For questions, use the Q&A function. The hand raising won't really do us any good because then we'll have to, it'll get very cumbersome. We'd have to unmute you and allow you to ask and blah, blah, blah. So if you use the Q&A function, I'll pass any questions along to Tim and Alicia. And with that, I'll just turn it over to Alicia and Tim. Thank you.
All right, thanks, Nick. And as always, this is because of the work through ILS that we are able to present this, but these are not the viewpoints of ILS. But we're here just to give just the basic understanding of DNA with respect to the collection of DNA and you know, some tips about the background of the science, and hopefully you guys find use of it.
So we're going to start with serology and blood identification. So with blood identification, it typically starts at the crime scene or at the search scene with a presumptive test. This is done by the investigator by the crime scene out in the field. They have something, they think it may be blood. They're not quite sure. They want to test it before they collect it. So they'll do a presumptive test in the field, and we'll talk a little bit more about that in a minute. And then there's a confirmatory test, which the evidence is collected. It's brought back in. It eventually gets to the lab, and there's a confirmatory test that is done that says, yes, indeed, we have blood here. And then the third test is the origin determination. Semen identification, it's close to being the same, a little bit different because steps 2 and 3 are combined. But again, there's presumptive test or tests that can be done in the field by the investigators, and then the lab will later conduct confirmatory / origin determination tests. So with blood identification, the first test that's done is, like I stated, the presumptive test. It's a chemical test which uses color changes to detect the possible presence of blood. It's a test that can easily be performed in the field by an individual, not necessarily someone with a science background. It's pretty much, I wouldn't say it's a foolproof test because there are a couple false positives out there. One false positive would be blood from a primate. Another false positive, believe it or not, would be blood from a ferret. Not that you run into too much ferret blood out on a crime scene, but it is, I guess it is a possibility. So with the chemical test, you get a sterile swab, which you can see on the left-hand side, and you put some sterile or distilled water on it and to moisten it, and then you rub a little bit of the stain in question with the swab, enough to just transfer a little bit of color onto your white cotton swab. And then it's treated in one of two ways. It's treated with a chemical, which you can see a combination of chemicals there on the left-hand side, or there are strips that you can buy over the counter at a pharmacy. The same strips that individuals would use, say, for instance, a doctor suspects that you may have blood in your urine or wants to determine whether or not there is blood in your urine because there is something physiologically going on inside an individual. So he wants to detect for blood. So the doctor may ask you to go to the pharmacy and get these little plastic strips that have a yellow end on the end of them. And what you would do is hold that strip, the end of the strip, in your urine stream. And if it turns a really vibrant, bright violet or purple color, that's an indication that there may be blood in your urine. This has been adapted to use at a crime scene or a search scene as a presumptive test. So you would transfer a little bit of color from your specimen in the field that you suspect might be blood onto your sterile swab, and then just touch it on the end of that strip. And within 3 seconds, if it changes color, then there's a good chance that it is indeed blood. There is a rapid determination, again, usually within 3 seconds. If it takes longer than that, then that could be a false positive. And on the right-hand side, you can, of this screen, you can see an example of sort of what it looks like. That's not a real test there. Actually, there's been a violet dot that's been added to this screen, but it gives you a good idea of what it will look like when you get that reaction. So the reason why I put this folded up pocket knife at the crime scene is to show you that when you're conducting a presumptive test at the crime scene, there's a determination that has to be made by the investigator on whether or not to indeed even do a presumptive test. There should be a large enough specimen contained on the evidence in order to allow you to do a presumptive test, plus still leave enough of the question stain so that confirmatory tests and tests of origin can be done back in the lab. So this would be a perfect example. In this case, I would think, and if I came across this piece of evidence, that there would be more than enough of a blood stain here to do a presumptive test. I would take one of the red dots in the very, very margins, the very, very outside portion of this stain, and just treat just that with, again, my moistened sterile Q-tip cotton swab, and do the test on that. So there are very many areas in which you can get a representative sample of the stain and still have more than enough left to do the laboratory tests. If there was only one little dot of blood, you would not do a presumptive test. You would say, I'm going to err on the side of caution. This could be blood. It would be collected, and it would be brought back to the lab where most, if not all of it, would probably be consumed in the lab in doing their confirmatory tests and tests of origin.
And Tim, if somebody is conducting, a law enforcement agent's conducting a confirmatory test, what's your opinion with respect to whether or not they should maintain those, like the swabs that they used?
I used to do it both ways when I did presumptive tests in the field, but I learned a very valuable lesson years ago, back when Samantha Zaldivar was allegedly kidnapped down in Wyoming County. I did the scene of her bedroom where she slept at night and had slept the night before of her alleged kidnapping, and found a large quantity of blood on an upper bed rail on the bunk bed. So it was dried, so I packaged it up in paper, and at that time, Monroe County Lab was doing all of the lab work for Wyoming County. Wyoming County did not have their own lab, so they contracted with Monroe counties. For some reason, and somehow, the lab destroyed the entire sample that was on the bed rail. So we thought all was lost, and maybe we couldn't figure out whether or not that was indeed Samantha Zaldivar's blood on that bed rail. But in that case, quite by accident, I had actually kept that presumptive test. I had kept that Q-tip, and there was more than enough of a blood sample on that Q-tip from my presumptive test to then bring to the lab, have analyzed, and we realized that indeed it was Samantha Zaldivar's blood. So after that, I always routinely kept my presumptive tests and logged them into evidence.
No clear policy as to whether or not you should do it or not, though.
No, there is not. But good question to ask, especially when there's just minute quantities of the suspect stain.
Yep.
Then with blood identification, you have your confirmatory test. It's a crystal test which uses the formation of crystals to indicate the presence of blood. And right now onto your screen, is, lo and behold, has appeared what it looks like with a crystal type test, what it'll look like under a microscope. Semen identification. The presumptive test in that is, first of all, there's a chemical test, again, which uses a color change to detect the possible presence of semen. And then there is also the use of the alternate light source, which I think is even a better way to look for semen stains, especially if they're possibly old semen stains, diluted semen stains, very minute portions of semen, because using the alternate light source, using a blue light and orange goggles, the stain will literally just jump right out at you, no matter how old and diluted, whether it's been laundered several times, it'll just jump right out and be very, very obvious. With the chemical test, first you have to find what you suspect is a semen stain and then conduct the presumptive test on it.
Hey, Tim.
And again, yeah. Yes,
There's a question. When they do the microscopic views and they're looking at what they look at, are there photos taken of the microscopic view itself?
there should be. And again, that happens outside of what I do or used to do as a crime scene investigator. I was basically a find it, bag it, and tag it guy, and then it went on to the lab. But typically, when these cases are all analyzed and they go to the lab and everything is looked at, it's always looked at in the light that this case would someday go to trial. So it would be better just to photograph it right up front when it's being looked at for the very first time, instead of going back. With rare exception, there are some rare exceptions where there are some chemical tests where, as time goes on, it may look one way when it's initially done. One good example would be latent fingerprints treated with ninhydrin. As soon as they're treated with ninhydrin, within a minute or two, you're going to see a lot of latent fingerprints on a piece of paper or on a paper envelope or any other porous type material. But 30, 60, 90, 6 months later, that chemical reaction is still going on, and you can pull that piece out of evidence and there'll be even many, many more latent fingerprints than the original latent fingerprints you saw will actually be even more vibrant. So it's one of those things, it depends, but they always should be photographing as they're doing confirmatory testing. So back to the alternate light source. Again, that is the second presumptive test for finding semen. It could be some other dried body fluid. Saliva fluoresces just like semen. Dried urine fluoresces just like semen. Blood does not fluoresce. Blood absorbs every wavelength of light. So no matter what wavelength of light you hit it with, no matter what color goggles you have on, the blood will appear very, very dark. In some cases, it'll cancel out the background. So when you're looking at blood through and using an alternate light with goggles on of one color or another, it may wash out the entire background so that all you're left with is the blood. But the blood will never fluoresce. Remember, fluorescence is an object taking in the light waves and then immediately radiating them back out again. And that's what you see through your goggles, what light wavelength is being radiated back out again. All right, next slide. And then you have your confirmatory origin determination test. This is an example of what one would look like. It's an antibody reaction test, which indicates the presence of human semen through the formation of precipitant bands in two locations, the test line and the control line. So if you look in the middle of this slide, the very first one, your sample goes in the bottom where it says S. So you would transfer a little bit of your stain onto the sample. That stain would migrate up this test strip. And if you get a bar across the C line, that's your control line. That lets you know that the test is working. And the T line is the test itself to see if it's positive for semen. So on the left-hand side, the test strip, you can see the test was working, but it is negative for semen. But on the right-hand side, where it would be applied, you'd have both the control line letting you know that the strip is working and the test line filled in. So this would give you a determination that this is positive for semen. So where is DNA found? DNA is found in the cell nucleus, only in the white blood cells, and nuclear DNA analysis is the most common, but there's also mitochondrial, which also contains DNA, and that analysis is useful with hair bones, teeth, and burned or charred remains. So this particular screen is, this slide is sort of an exploded view of a cell and what it looks like. And by the way, this is where I say that I'm not a scientist. I was a crime scene investigator, so we can't dive too deep into the science in this area. But just to give you an idea of what's being looked at, this is an exploded view of the cell's nucleus. So you have the chromosome where there is the compacted DNA, and it is basically unwound into the double helix where then it's uncompacted. And then you can see the target region, which is used for the DNA typing. And I believe Nick and Alicia, everyone's going to get a copy of this or have a copy of this
Yes,
slideshow available to them.
they all got it as a part of the written materials.
Right, so this is something you can review down the road again, especially down near the end of this presentation. We're going to have a few slides that are full of a bunch of information, and we're going to go over it superficially, but it would be nice as a reference or a refresher down the road if you have a case involving DNA. So mitochondrial DNA, most useful, again, like I stated, for the identification of skeletal remains, burn remains, or dried bones. Used as confirmation after a visual hair identification. It's maternally inherited, so it's, in that case, it's the same in all siblings, and the mother, but different from the father. And it cannot provide ID of a specific individual, but it only matches to the individual's maternal line.
And Tim assured me that this photo is not from a crime scene, but like during doing research at a body farm. So there was not an officer there making light of investigating at a crime scene and then us using this photograph. So
absolutely, I can just tell from the photo that it was at one of the body farms that the FBI conducts classes and research at, and or if it was a real thing, yeah, I would hope he wouldn't be smiling like that and posing with a skull, and he would be fully decked out in all his PPE. So sources of nuclear DNA, and I'm sure you can read the slide: semen, sweat, saliva, vaginal fluid, blood. I've also put in here urine and feces. While in both cases you will see that it states that it is not your best option. It is not usually even looked at. It's looked at as a sample or a source of last resort. There really needs to be something physiologically going wrong with an individual for them to have DNA or quantifiable amounts of DNA in their urine or feces, which would maybe possibly they're bleeding internally, which would cause that to be there. So not your best options, not usually looked at in the lab, or for that matter, even collected in the field for DNA sourcing. Typical items with nuclear DNA: clothing, eyeglasses, hairbrushes, baseball caps, cigarettes, drinking cups, which you'll hear about investigators following individuals around till they discard a bottle or a drinking cup. Stamps, when we used to have stamps that you would lick, the flap on an envelope, and things of that nature. So DNA, what is DNA's downfall? What can damage DNA? For one thing, sunlight, prolonged exposure to sunlight can destroy DNA. Prolonged exposure to a high heat situation can destroy DNA. And moisture. We've talked about moisture in previous presentations we have, but high or prolonged moisture is not a friend of DNA. It can cause mold, bacteria. It can cause your sample to rot, putrefy, making it useful at worst, and will degrade it at best. So I mean, you could still possibly have some degraded DNA to work with, but if for all intents and purposes, it should not be exposed to moisture for any length of time. And there's some ways of preventing that from happening, which we'll discuss. DNA may survive laundry procedures, fingerprint powder, and superglue treatments. So laundry procedures, let me give you an example of how resilient DNA may be, for lack of a better term. It has been studied in the lab, and I have seen examples of DNA that's been diluted down to 10%. So what we're talking would be, say, with semen, 1 part semen to 10 parts water. So you've got a dilution of 1 to 10. And I've seen that 1 to 10 dilution laundered 10 times. And it is still very, very visible using an alternate light source. It will just jump right out at you and glow on like an undergarment or anything else that it had dried to. So it can survive dilution. It can survive laundry procedures. It can also very easily survive fingerprint powder. And if fingerprint powder wasn't used on latent fingerprints and it was taken back into the lab or processed in the field for superglue treatment, it can even survive a superglue treatment, because while the superglue will affix itself to the latent fingerprint or to the whatever the stain is containing the DNA, it will not destroy it. So it should never be assumed that there can't be any DNA or there will not be any DNA that survived.
And I think that that's an interesting point with respect to, you know, why we are doing this remotely right now, and the similarities of the transfer of a virus and the transfer of DNA. And I think that, you know, as many things have now changed, once we can get back in the courtroom and once we can start having jury trials again, we're in a very unique position where, like never before, jurors are going to have a better understanding of transfer, and they're gonna really get it more than they ever did before because of what's going on now. Because, you know, no matter what they're doing, it's hard to avoid the conversation or the dialogue in the news every night where they're talking about the ability of coronavirus to exist on an Amazon package or on your shopping cart or your groceries, and if it does exist on those things, for how long it's considered a living virus, and your need for wearing gloves and our mask and everything like that. But that's where, you know, these people are experiencing it and they're talking about it being a living virus and it being able to impact us. And you can come in and, you know, remind them what that was like and how they understood that somebody who is sick touching something else and you know, another person, healthy person picking it up could become ill from that. But that the major distinction is that virus eventually dies, whereas, you know, DNA, while it can be degraded, especially from like the sun or heat or anything like that, but technically its existence can exist indefinitely. So while they understand this idea of transfer more, you know, even pushing that DNA can exist even further, you know, especially under the right circumstances, that it can continue on, I guess, and keep and exist there for forever. So with the transfer in mind, DNA TPPR, which stands for transfer, persist, prevalence, and recover. Transfer being the ability of DNA to actually transfer directly or indirectly. This can be impacted by washing your hands now, but its actual ability to transfer from one individual to another or to an object. Persistence, DNA in its ability to persist on an actual item or object. And there's a lot of studies on all of these different issues. This in particular, they've done studies on items that are used exclusively by one person, and then, you know, presumably used in a crime, or if something would have been returned to somebody who is a consistent user, how long that short-term use would impact the DNA on the object, like thinking about a kitchen knife that somebody is using regularly and then for some reason is used in a crime, but for some reason nobody knows about it being used in a crime, and it is returned and it's continued to be used the right way. There are studies about the persistence of the DNA on items like that. Prevalence, the commonness of DNA to be present on an item, studies about non-self DNA on people's hands caused by shaking hands. They've done studies on mothers' necks who, you know, have small children and hold their kids, and their kids are always grabbing at them. What is the prevalence of having another person's DNA on their neck? And in the case that Tim was mentioning before about the laundry, you know, these child abuse cases and the existence of semen DNA from semen on the clothing of children, and experts coming in and they're trying to really just distinguish background from a deposit, as well as the recovery plays a role, the sampling, and how that'll make a difference with respect to how much DNA you're getting, or what you are able to get off of a certain object. If a swab was used, cutting, wet vacuum, however it was recovered, depending on what it is, would determine whether or not they're getting a sample off of it. So these tables are, you know, examples of primary transfer and indirect transfer. I won't go through every single one, but the idea of, like me, I'm holding this pen in my hand, and presumably my DNA is coming off of my hand going onto this pen. And then I put the pen down on my desk. The DNA that I have left on the pen, it can transfer from the pen onto this desk. And if somebody else comes into this room, and whether it is them picking up the pen and getting my DNA on that person from the pen, or maybe after I leave, somebody comes in here and touches the desk and they're getting my personal DNA off of the desk, which came from the pen, you know, there's this whole big line. And, you know, getting these concessions out of the state's experts about the reality of transfer and about all of these studies, and, you know, a big concession about the fact that, like, timing of DNA of DNA transfer is unattainable. They can't say, when they test something, when they look at my pen and they find my DNA on it, they don't know when my DNA was placed on that pen. So when they talk about that, and then you make them testify objectively and not subjectively about transfer, and you ask them about their familiarity with DNA transfer, and they say, of course I'm familiar with it, and that's how DNA gets from one person to another person or to another object. And they say yes, and you're also, Mr. or Mrs. whomever, you're also familiar with secondary transfer. Yes, I am. And that is where DNA is transferring from a person to a second person, and then from that second person to either a third person or to an object there. Yes, it is. So it's fair to say that it can transfer from one person to an object, and then an object to another person. That's another form, correct? Yes, it is. In general, secondary transfer relates to finding a person's DNA on an item or on a person when they never actually came into contact with that item or person. Yes, generally, yes. So we're passing a microphone, you know, we're passing a microphone around during jury selection. And because of that, if the microphone was now swabbed, you wouldn't be surprised to find my DNA on it or any of the jurors' DNA on that microphone, because we all handled it. It was passed around between all of us. That's fair to say. Well, what if one of the jurors, prior to handling the microphone, was out in the hallway, ran into an old friend of theirs, shook their hand, and then came in and handled that microphone? it's possible that that friend's DNA would be on that microphone, even though they didn't come in, they never actually individually handled that microphone. And that's an example of secondary transfer. And I think that's a good way to put it, in that, you know, it gets jurors thinking about the fact, something that they've been involved in, something that just happened. And if you got a gun case, it helps the fact that it's, you know, small, dark, black object, weighted object. So I think getting into the issue of transfer that way, and just the objectively, prior to actually getting into the case and getting your concessions in before you're getting into in depth on the facts of the case. There are a lot of different factors that can affect transfer. Substrates, that's the types of surfaces, can impact how much DNA is transferred between objects. You know, the different surfaces such as glass or wood or a tape is more likely to collect more DNA than something else. Biological material, you know, body fluids, they obviously transfer more easily. If it is a wet body fluid, more easily transferred than a dry one. If it's fresh material, it's not degraded by anything, you know, more likely to be more transfer. And the source of the skin of where it's coming from, different areas of your hand are more likely to transfer DNA than other areas of your hand or body, which goes into shedder status. You know, studies have shown in their studies about all of these different factors that affect transfer. The studies show variation in the propensity of individuals depositing their DNA upon contacting an item. I think it's important to get out that this is, you know, obviously depending on your case, the facts of your case, getting out the fact that this is something that exists out there. It's tough in that, you know, you get it out there and you say that this is a thing, and, you know, people shed more DNA than others, and that could be why somebody else's DNA isn't on there. But there is no real test to say, oh, Alicia, Lilley, we tested you, you're a shedder or you're not a shedder. So there's no way of sitting there and having a defendant or a co-defendant, your client, tested to say, you know, how much DNA they are actually shedding. It is a concept and something that does exist. The manner of contact can obviously impact how much DNA is being transferred from a person to an object, or an object to another object. How long I am holding my pen, how I held it, the length of time. Force or friction can impact transfer. It's been found in sexual assault studies, or sexual assault cases. You know, the more force, the more friction. There's been shown that there has been more DNA transfer in those circumstances.
So the actual DNA evidence collection, we've touched on, you know, considering these possible cross-contamination issues, and that can extend to the investigators at the crime scene, or whoever collected the evidence. Were gloves worn? We've talked about that in previous meetings that we've had. And did the investigator change gloves appropriately? You know, when they're moving from one piece of evidence to the next, one room to another, one area to another, did they do proper glove changes? Was Tyvek worn by the investigator? Did investigators clean their equipment between crime scenes? That's an area that a lot of investigators never even considered. You know, were the investigators at a crime scene a week earlier and setting down their crime scene equipment and their crime scene kits on the floor or on the couch at the location they were at? And did they then pick up some DNA and bring it with them to another search scene, another crime scene? Did investigators use those one-use supplies that we've talked about in a couple different presentations now, previous presentations? So are they using the one-use supplies? The supply where they're using the fingerprint powder once and then throwing the rest away, using the fingerprint brush once and throwing it away. Those issues where there's a possibility with your equipment and supplies for cross-contamination, were they used just once? Was the entire item collected? Was there a lot? Was there a big stain? Was there a large item? Was it an immovable object that they had to collect a sample off of? How much of it did they collect? Did they do a proper representation of the entire stain or of the entire object that could or did contain DNA? And again, were those samples collected from different areas? You can't assume that if you have a bunch of blood spatter on a surface or a trail of blood across a room, especially if there's intersecting trails of blood across a room, that it came from one source. So was there the proper collective sample taken from different areas?
And I think, you know, if transfer is an issue in the case and, you know, we're really going to go after the DNA, these are things, this is where it's got to be developed first with those law enforcement officers and whoever collected the evidence. And, you know, I think I said once before about the idea that sometimes we stipulate to some things that don't seem like a big deal, but I think if you get into it in greater depth with your officers and detectives, you're going to have a better argument, I guess, with the experts. Whether it's your expert that you're calling, or, you know, just during your cross with the state's expert, because of I think that a lot of them, and like I've seen them concede issues of transfer before. So when you get them on just the basics of the theories, and then you get more case specific with respect to these issues, you know, about the investigators not cleaning their equipment. If you talk to the investigators about the fact that they're not cleaning their equipment, or they're not using one-use supplies, and they've been a detective for over 20 years, and they happen to have used fingerprint powder that they've never replaced, and there's very little corroborating evidence on your case besides your client's DNA where the fingerprint was taken. And, you know, maybe we don't have a matching fingerprint, but we have their DNA present there. Going in with the officer and getting out the fact that how long that's been around. We've got the concessions from the expert with respect to transfer in general, and then going in more specifically about the fact, like, isn't it true that DNA could be picked up, you know, from an object such as a fingerprint brush? A brush would pick up DNA, correct? And that brush could then, if not properly cleaned, could transfer onto a different object. Or getting out with your, you know, the gun in the car case, getting out the fact that DNA can be found on the rubber gloves. You know, you put on your rubber gloves, they're no longer sterile anymore. Once an individual starts touching things with the gloves, they're going to be collecting DNA from whatever they're touching. And if an officer put their gloves on and pat down your client and then began searching a vehicle, or then recovered the item of evidence that they had stored there, then, you know, you'll get those concessions with respect to the fact that that, you know, the DNA could have been collected on those gloves and then put into the vehicle or put into the item of evidence. Or maybe they put on their gloves and they looked in a backpack or went through a sweatshirt, and then they find the gun, you know. and getting out the fact that those are possibilities for transfer. And the idea about collecting the entire item of evidence or the entire sample, you know, did they miss a sample? Did they not collect something that they should have collected? Because, you know, it only takes one allele to possibly exclude somebody. And if they didn't collect everything, if they didn't take everything, then how do we know that that's not the sample that would have exonerated your client? So I think that those are all things that you can bring up. I totally forgot to insert the code. I am so sorry. So I'm writing one right now, and I have awful handwriting. It is ABC 123. Can you see it? Is it backwards? It's backwards.
Good.
All right, ABC 123, and I will write another one. I'm really sorry, everybody. Totally forgot. So that's what I got.
Just to touch on that one more time, and not to beat us to death, but just another example. And these are all thought-provoking examples we give. But I mean, years ago, if you're using fingerprint powder at a crime scene, and you powder them, and you saw some great latent prints that you had developed, you would lift those and put those with lifting tape or by some other means, a lifting gel, and place them on a card and submit them. You'd have other ones where there was a smudge where, you know, maybe the individual's thumb turned after making contact with an object. So you had a latent there, but it was not an identifiable when you had a smudge, and those wouldn't be collected. Where everyone hears about touch DNA, where it could be really, really light and unidentifiable. So, you know, the days of not collecting those are over, because they still have value now. That smudge, while it may not have a latent fingerprint value, it does have a DNA value, you know, as does, of course, the touch DNA. So imagine someone going to his kit and pulling out a fingerprint powder jar that he's had for 5 or 10 years. And let me tell you that a jar of fingerprint powder will literally last 5 or 10 years. And a good camel hair brush is very, very expensive. They can be as much as $30 or $40. And someone has, you know, their go-to camel hair brush, their tried and true brush that, you know, develops latent fingerprints when no other brush works. And, you know, it's their favorite brush that they've used over and over again at hundreds of scenes. Now, if they're processing that smudged print or that very, very light, unidentifiable print with their powder that they've had for 5 or 10 years and their camel hair brush, which they've had their entire career, imagine all the cross-contamination there is that exists in that powder and on that brush. Packaging. So to survive, DNA evidence should have been kept away from, and we had this in previous slides, sunlight, moisture, and heat. So how do you do that? Typically, DNA evidence is always packaged in paper or some type of paper breathable container. Cardboard is in the same family as paper. Or you would use another breathable material such as Tyvek. Tyvek looks more like a plastic material, but it actually does breathe. And there are Tyvek envelopes, crime scene envelopes that are made that do breathe. So you actually could put something in it that contains some moisture, and it would naturally dry. Wet evidence. If you have wet evidence at a crime scene, it should not be packaged or only packaged loosely and temporarily, and it should be brought back into a controlled environment in the office or in the lab, where it is then thoroughly dried, either naturally by laying it out on a surface, a clean and dry surface to dry, or it should be placed in a drying chamber before packaging. A little bit more sophisticated technique, but a very, very good technique, and a technique that accelerates the drying process tenfold. So again, if they have wet material, if they are bringing it back to the office or the lab where it's a more controlled environment and where it can dry, was the surface that it was put on cleaned, sterilized beforehand? Was the drying chamber cleaned, sterilized before evidence was put in it? Again, another opportunity to consider possible cross-contamination defenses. So if you come across a case where there was wet evidence, you need to explore how it was treated after it left the crime scene.
And I think, you know, if evidence isn't being stored correctly, you know, that it's not packaged correctly, the idea of the destruction of that evidence because of, like, that could result from that whole idea about, you know, they didn't collect all the evidence, they didn't collect all the blood that was there. Would that have exonerated somebody? The same thing, they damaged something, the chances of exoneration from that, or you know, just adding another suspect. If they did that wrong, what else did they do wrong? That whole idea of, like, we learned the right way, the best ultimate way for them to do everything, so that we can highlight the things, you know, that, like, how they're not doing it, and in exploring all of the cross-contamination issues as well.
So I added a slide here to show you what a drying cabinet looks like. Most of you have probably never seen one, or for that matter, ever even heard of one. They do exist. They are becoming more and more common. This is the exact replica of the drying cabinet I had at FBI Buffalo, and actually every FBI office, field office in the FBI has this drying chamber. I know Amherst has a drying chamber. I know the state police have drying chambers. They're out there, they exist. I don't know, I've never seen one when I was over at Buffalo PD. They may or may not have one, but the concept of how they work, they're just a very, very large and sterile closet, so to speak, that you can place evidence in, either on shelves or hang. You'll see in the front of them, those are plexiglass windows. In the lower part of those plexiglass doors, you'll see those rectangular plastic areas with the 4 knobs on it. That's where filtered air comes in. There's a pre-filter there that'll filter air coming in. There's huge fans in each one of these devices that'll pull the air in through those pre-filters up through and across the evidence, and then the air comes out, exhausts at the very top after it leaves about a 6-inch high HEPA filter, which takes out all of the impurities and from the air and the smell, in case you have a very smelly piece of evidence. So these machines are very, very effective. They dry the evidence at a very accelerated rate, but they must be used properly. In between each use, they must be fully and totally cleaned and sanitized. And there is a procedure to do that, and it literally takes hours to do. This machine, these closets actually come with a compartment where a hose pops out, and you actually connect this to a water supply. They are hosed down with a disinfectant, and they're in the bottom of these cabinets, these closets. There are drains, and then the water is drained out, and then they are locked up, and a green zip tie is placed through each of the handles. Each of those 2 handles you see midway up the doors has a hole in them. They're locked with a green zip tie, green for go, which means that they've been disinfected. And then the unit is turned on where it is, and it is thoroughly dried inside. If you come across the drying cabinet and are thinking about putting evidence in it, and you see a red zip tie on it, that means it has been used. The evidence has been now removed because it's been dry. An individual or individuals did not have time to decontaminate the drying cabinet, so they've placed a red zip tie through it to signify that it is dirty. It does need to be decontaminated before it can be used again. So again, just one more thing I want to, and you're dealing with evidence that at any point in time was wet, and it doesn't have to be clothing. I recovered a handgun once from the back of a toilet bowl where the bowl reservoir was. I lifted up the lid and there was a semi-automatic 9mm handgun in there. Took it back, used the right side of the cabinet where you can see there are shelves, just carefully dismantled the gun, placed it all in there and dried the gun out so that we can then conduct latent fingerprint analysis on it. So it's a very effective piece of equipment if used properly. So DNA standards, what are the knowns that you use to get DNA from? You have whole blood sample that is typically collected in a purple top tube, and it contains an anticoagulant that protects the DNA's integrity. Buccal swabs, some people pronounce it buccal swabs, it's buccal swabs. They are contained in a DNA collection kit. They look very, very much like those previous slides we saw where there were the sterile cotton swabs. Those swabs are run on the inside of the cheek down into where the cheek meets the gum line. That's where DNA cells collect in the most abundance, and that's typically rubbed vigorously to transfer some DNA onto the buccal swab, and then those would be sent in for analysis. And if the victim is missing, then you can get DNA standards or DNA knowns off of their known toothbrush, their hairbrush, eyeglasses, razor, and you can see the other ones, cigarette butts, etc. Types of DNA analysis, and we're going to hit on this superficially. Again, Alicia and I are not scientists, but we'll give you a little bit of the different techniques and testing out there and the history of it. So we start back in the early 80s, in and around 1983 with RFLP, which was the first widespread DNA profiling technique. It's now largely obsolete due to the newer and more inexpensive sequencing techniques. The reason we bring it up is in case any of you are involved in cold cases of any type that date back to the early 80s or all through the 80s and even up into the early 90s. You may come across the first techniques used on the evidence was RFLP. PCR is a technique. It's multiplying copies of the targeted area of the DNA many, many times over so that it can then be analyzed. And STR, that's what most people are aware of today. It's what's most prevalent today, the short tandem repeat. It's the current method, of course. It compares the length of different sections of the DNA chain. It's similar to RFLP, but it can use much smaller samples that are replicated because it uses a PCR-based technique. So PCR, we have a little diagram here on the left, which just sort of gives you an example of the type of machine that's used and what the process looks like, but it's the technique that is used in multiple typing methods like STR. It enables the use of a very, very small stain, much smaller than previous techniques. So if we go back to one of the original slides we had today where you might have that pocket knife, which maybe only contained one tiny little stain, you could still replicate it several times over. When I worked at World Trade Center, in some cases when we were looking for remains of individuals that perished at World Trade Center, we would sometimes find very, very tiny amounts of DNA, so small and and so degraded that we would have to put it on a slide and looked at under a microscope. And in that case, the New York City labs actually developed a new technique called laser ablation, where they would actually see a cell on the slide and shoot it with a laser beam from underneath and pop off just one cell, which they can then multiply over and over again using PCR to try to identify those human remains. This just gives you an example of what we're talking about. So, you know, instead of our small sample, we have, you know, bunny rabbits here, where you start out with one, and then it's basically molecular Xeroxing, where you get one in your targeted area, and then you just multiply it over and over again to get identical copies. Where it's used: research diagnostics. And we move on to our STR, short tandem repeats. And again, this is just a slide to give us an example, or to tell us where it's best used and why it's best used. It's PCR based for getting a small sample and making multiple copies. It's good with low quantity DNA. It's good with testing degraded DNA. It's highly discriminating. It was expensive at one time. It's not that as expensive anymore. And it's amenable to automation, which is great. It's an automated process now. It's a process that can be conducted over a relatively short period of time. So this slide here, we debated whether or not to put it in or not, how deep into the science we wanted to get. We just threw it up here again, just to give you an idea, a picture in your head of what we're talking about with alleles. We're not going to discuss alleles too much here. Alicia will talk about it a little bit more in upcoming slides, but you basically have repeats of the alleles. You have mom's alleles, which in this case is designated by boxcars being towed by a train engine. And you have the mom with 3 repeats, and you have the dad with 4 repeats, or boxcars in this case. So when we talk about the alleles, without getting too deep into the science, just kind of think of this representation that we're giving you here. An example of what an STR profile looks like. Again, as we stated, it uses the PCR technique to reproduce the sample, and again requires only a very minute trace of DNA, which can be replicated over and over again. And if there is a match, the lab provides the probability of selecting an unrelated individual at random who has the same genetic information as the crime scene sample. And the probabilities generated can be more rare than the previous techniques that existed for profiling.
So when we're looking at DNA, you're going to see a huge difference, obviously, between single source DNA and in comparison to a mixture, and even a huge difference between the mixture of 2 individuals versus 3 or more. It's obviously getting more complicated. And now that we deal with touch DNA so much, we do more of it, you're dealing more with mixtures because that DNA, it's coming from a variety of different sources. It's not necessarily just contact, it can be secondary, it can be tertiary. So there's a lot more being mixed into the sample. So, and you can see both things on an item of evidence, like a knife used in a stabbing. The swab of the blade may be a single source. It may be really easy to interpret, really easy to compare with the known sample, as opposed to the handle, more likely to be a mixture if it's, you know, if a knife that was possibly used by somebody who's not the owner of it. There's going to be more peaks with that comparison, and there's the potential that the peaks would be all different sizes. So here is an example of a single source comparison. It's very easy to read. It's easy to interpret. Dr. John Butler, who's considered the godfather of DNA evidence, he is also the author of several books that you will find in the library at the assigned counsel's office now. I've written about his books. He said that you have basic math is, 2 + 2 = 4. That's the basic arithmetic. And that's the equivalent of doing a single source comparison. When you get to a sexual assault evidence, 1 perp, 1 victim, the DNA is mixed, that's algebra. High levels of DNA there, it's not really an issue. Touch DNA, he describes as calculus. And when, the idea of like talking about DNA and the gold standard, it's not the calculus that they're talking about. It's not the mixture or the touch DNA. The gold standard is this single source comparison or the simple mixture. Here is a simple mixture from, say, a sexual assault case that, only had 1 perp. The victim hadn't had any sexual contact with anybody else beforehand. It's easy, it's uncomplicated, there are high peaks, it's pretty easy to read, easier to identify. Here is a close-up of that with the evidence on one side, the known sample there. This is our algebra when making the comparison. When we get into the complex mixture, and I just have the close-up photograph of it, this is our calculus. You can't tell which peaks pair. You have to take into consideration that different peaks may pair up. You're going to have lower samples. It can cause, when you have a complex mixture, you have a lot of different things happening there where the peak height may be imbalanced from sample to sample. It should traditionally be the same height in each different sample, but that might not happen in the mixture. So you don't necessarily realize that it belongs to the same person. You'll deal with dropout more, where the test just fails to pick up an allele altogether. Drop in, which is a sporadic contamination, where alleles flow from the air and get in there. That's pretty uncommon, but it's a possibility. Stutter is the pretty much where the PCR process loses its place, and you'll get a peak either before or after the peaks along the electropherogram. This stutter can actually happen quite frequently, but in a complex mixture, it's much harder to identify it. So that becomes an issue when looking at a complex mixture, as well as stacking and sharing. If multiple people who are in the mixture share an allele, they're going to stack on top of each other, and it could result in there being a very high peak, and you can't see who it's coming from. So those are issues that come in with mixtures. And I'm just gonna, sorry Tim, I'm just gonna comment a little bit more about the mixture that, the complex mixtures that SAR mix and true allele. They're proprietary algorithms. They are there. It's a probabilistic genotyping, and it is meant to unmix the mixture kind of and place it in individual parts for us. It compares different combinations of contributors. It's trying to make the best combo for us. It tries to analyze the evidence, I guess, explaining what we're seeing in the electropherogram, and then it will also make adjustments for data that is missing. It makes adjustments for uncertain data, and then it applies the likelihood ratio. So it's a lot better than what has ever been there before. Scientists looking at it in and of themselves, it's better that it's this system, but it's not foolproof. It has to be calibrated. There's the human element to it. They have to understand what they're doing, but it's something that's designed to help them. But it's certainly, the more now with new discovery, we should get more background with respect to these these programs and these algorithms, but I don't know exactly how much is being turned over. But I know I've spoken to several experts that are very much looking forward to looking at all of the new 2020 discovery. So I have the second code for you. I don't know if you can see it. It's SFL 729, and I apologize that it didn't go on a nice, pretty slide. So that was totally my fault.
Ready?
Yep, sorry.
That's okay. So we're getting down to our last few slides here. We wanted to touch on, for just a few minutes, the National DNA Index
Oop.
System.
I don't know what I just did. I'm sorry. There. Sorry.
So the National DNA Index System, NDIS, is part of CODIS. I'm sure you're all somewhat aware of CODIS. CODIS contains the DNA profiles contributed by federal, state, and local labs. And I've got this representation of it because I think the best way to try to understand how CODIS works is that it's like a bucket that contains each, and with each bucket containing different categories of samples. So there is a lot of indexes that that make up CODIS, and rather than try to put them all in a slide or in slides, when we get you out this PowerPoint presentation, we'll make sure that enclosed in it, Rachel gives you a list and a definition of all of the indexes. It'll either be contained within the PowerPoint, or I have it on a separate sheet of paper, which I'll make available to Rachel, and she can attach it to the emails to all of you. But I wanted to go over some of these indexes contained within NDIS and part of CODIS, just to give you an idea. Some people think that there's just this one giant CODIS out there and all the DNAs are just all lumped in there together. That is not quite the case. I mean, they are all in there, but they're by separated by an indexing system. So you have to think of CODIS as a system of pointers, and these pointers are all housed in the various indexes. CODIS index is analogous to an index in a book. It simply points to DNA profiles created and managed by the various state and local forensic labs. So to give you an idea of some of the indexes that are contained within CODIS, there is the forensic index, which is a match of forensic indexes associated with cases, with other cases nationwide. And this helps different jurisdictions coordinate their investigations. There's the convicted offender index. A lot of you are probably aware of that. Crime scene DNA profile is also searched against these, and suspect DNA that they have is matched against convicted offenders that are in this index. Then there's the population file. They call it the population file. It's actually another index. It has DNA profiles that have been highly individualized and are contained in there, so that you have entire populations that can be compared against when creating these percentages and these ratios. There's another index called the unidentified person index. Okay, so this has to do with the DNA missing person database. So when you have missing persons, their DNA is obtained and put in there for possible future use. And what they do is compare, say human remains are found, and they'll extract DNA from those human remains and then go to the databases and see if if a loved one, a family member, has reported the person missing and has actually given their DNA to compare in this database against unknown persons that are found. This is sort of what I refer to as the civil side of CODIS, the missing person DNA database. So you have donors that are family members that are donating DNA. You have human remains that are found at some point in time. DNA is extracted and compared against the donors to try to make matches and to help loved ones and families get some type of closure and wondering what happened to loved ones that disappeared, whether they be runaways, people that may have been kidnapped for whatever reason. And there's also another index out there called the Victim Index. And these are victims of crime that have, say in a rape case, that have voluntarily submitted or provided their DNA, and it goes into this database to help maybe solve their unsolved crime in the future when maybe a serial rapist has been identified. So all these different indexes exist within CODIS, and we will provide you with a list of them and their definitions. So CODIS, it is made up of samples from federal, state, and local agencies that are all working together to create this giant database. It maintains the profile of both crime scene samples and convicted offender samples, as I talked about, in those two different indexes. Samples are automatically cross-compared on a regular basis to try to make identifications. Also, like I said, the civil side of it contains missing persons, relatives of missing persons, and unidentified human remains. And that is that missing person DNA database program that is sponsored by the FBI. So this is the architecture. This is the way the NDIS works. You have at the top the United States, or in this case, it's the FBI that oversees this whole NDIS system. Then you have the state laboratories that are providing samples, and below that you also have your local labs across the country that are also supplying DNA profiles. So all in all, there are 190 sites in the United States with the FBI overseeing the program. 136 of the 190 are local labs, and 54 are different state labs. So this is tried to give you an idea of how CODIS works, and it's extremely convoluted. There's a lot to read here. We aren't going to read it now. We are providing you with this PowerPoint presentation. It's something that you can refer to in the future if questions ever arise with you in your cases regarding CODIS. So I provided this to you, and hopefully it will make some sense in taking you through the step-by-step process involved in deciding which index within NDIS the DNA profile might be, when is CODIS used, when it is not used.
All right, I'm here. I'm going to turn myself on. So a couple questions came in throughout. I decided to just leave the rest till the end. So anyone who doesn't have a question and doesn't want to stick around, you're free to go. You're going to get the 1.5 hours of credit anyway. But if you have questions, we had one early on, Tim, when we were talking about the testing for the presence of blood. And the question is, if a presumptive test comes out false negative, does that close the door to going on to the next step and doing further testing?
No, not necessarily. And I mean, there are probably there are cases out there when I got an indication that it wasn't blood, and I mean, I was so sure that it was blood. For example, I'll go back to the case I gave before, the murder of Samantha Zaldivar, the 9-year-old child that was allegedly kidnapped and murdered by her mom's boyfriend down in Wyoming County. Among the other evidence I found in her bedroom, I found what I was absolutely positive was a bloody handprint of either Samantha's or her younger sister, and it came up negative to my presumptive test. But I was so positive it was dried blood, I thought maybe there was a problem with the test itself. So I collected the sample anyways. It was a bloody print on wallpaper in the bedroom, so I actually had to cut the wallpaper out of the wall to submit it. Lo and behold, it was not blood. It was cosmetics that the kids had gotten their hands into and made this print on the wall, and it looked exactly like dried blood to me. So I collected it anyways. So it's not a be all to all. I mean, it is just presumptive. You could still collect it. That's basically one of those things that you decide at the crime scene.
Okay. Alicia, if you're trying a case and your idea is transfer of DNA, when do you first start to raise that idea during your trial?
Well, I think you're doing it with the officers, and that you are getting into, really in depth with them with respect to what they were doing, in their collection of evidence and everything that they touched beforehand. And, you're really paying really close attention to detail with them, and, in all the spots where they're not changing. If they're wearing gloves, everyone's always really happy to say that they put gloves on to collect something without ever paying attention to the fact that they keep them on and how long they wear them for. So really getting into detail about everything that they've touched and everything, and where they've been before they were at that crime scene, and their equipment and stuff like that. I don't think it would necessarily benefit you to talk to law enforcement about DNA. I think you just lay the groundwork with respect to the investigation, so that then once the state's expert is on there, you can get your concessions about transfer in and of itself, and then going into more in depth with your case before then having your own expert, especially in like a complex mixture case, if that's what you decide to do. But I do believe that the majority of the people at the CPS lab will concede most of these facts, although I understand that there's totally a possibility that somebody could stonewall you. So I'm hoping to start collecting transcripts of them, and especially where they've made good concessions, so that everybody could be equipped with those transcripts, so that if they're not giving you the concessions that you expect to be getting, that you can start crossing them with their prior testimony. So.
Oh, great. Okay. Tim, in the drying cabinet, do they change the filters every time? And if not, is that something that could contaminate evidence?
It's not likely that it would. The pre-filter is changed regularly. It's just in case you're in a room where it might be somewhat of a dusty environment, and it's more than satisfactory with pre-filtering the air. The HEPA filter in the top is good for about a year. Its main function is that the air that's exhausted back into the atmosphere doesn't smell. Some of this evidence you can have can be really, really smelly. That HEPA filter takes all of the smell out of it. There would be no way for the HEPA filter to contaminate anything inside the chamber. HEPA filter is probably around a $400 filter, and it's probably, based on usage, it's probably changed once every year or two.
Okay. Are there any distinctions by race or ethnicity in DNA?
I don't think it's determined by genes, so I don't think you'd ever be able to tell.
But they do, so on the reports, the preliminary report before the known sample is tested, they do usually say at least, do they sometimes break it down by race in those or no?
I don't think so, but I guess I couldn't say for certain.
Okay. So in looking at a sample, you can't typically tell, at least at our level where we are right now, what race or ethnicity the person is.
No.
Okay. No. Okay. What about, this is probably for Tim, what about the refusal to use different swabs for trigger on a gun rather than the grip, or for the barrel versus the trigger, barrel versus the grip? Is that standard in your line of work, or is it, would you say that's like an error in swabbing?
To use a different swab in all those different areas?
No, they generally, they will use the same swab in those areas unless there's a specific request from the DA's office. I think one of the attendees just wanted to hear your thoughts on that.
Yeah, well, I would think, to be safe, you know, a lot of times, I mean, you have a gun, I mean, you don't know where the case is going, and especially if, like me, a lot of times I would be just the crime scene guy. I wouldn't know all the background of the case, and sometimes you don't know what's important and what's not important, and whether or not, you know, the ownership of the gun, or what hands it went through, and who's cleaning the gun as opposed to who's using the gun. So I would think there is absolutely no downside to swabbing each area individually with different swabs. There definitely is a downside to using one swab for the whole gun.
Okay. And do they use the, this came up when you were talking about the different data banks like CODIS and things like that. Do they use that in missing persons cases at all?
Yes,
Okay.
they do. They do use it in missing person cases, and all that missing person DNA database and everything that goes with it, the unidentified person index, the victim index, not the victim index, yeah, but the victim index, the donations that are made to it. And again, like I refer to it as sort of like the civil side of CODIS. It doesn't cross over into the criminal, so it's only used for the missing person related end of it. So if you have human remains that are found and you extract DNA from, you have family members that are donating their DNA, you have missing persons where they've got known samples like we talked about in previous slides, and they've extracted DNA from that. That is only used in conjunction with resolving the missing person cases.
Okay. And then one more question, and it's for both or either one of you, I think. Do you know of any way, or I know they don't do this at the CPS lab, but is there any type of expert who can look at an unknown DNA profile and be able to tell you things such as the eye color, hair color of a suspect, things like that, based on the DNA that was found? Or are we not at that level yet as far as where the science is?
I don't think we're at that level. I think that those are all more like, I think they're like traits that aren't necessarily shown within the alleles that appear in our DNA testing. So I don't.
Yeah, or we don't know, you know, our science hasn't advanced to the point where we know where those genes are located.
Right.
In the chromosomes. Yeah, I think I agree with that. I've never seen or heard of someone able to do that. It would be great if they could. But,
absolutely.
Yeah. Okay. That was the last question. So thank you to all our attendees, and obviously thank you to Alicia and Tim for sharing your knowledge and expertise with everyone. Make sure you send in your evaluations and your verifications. Any code issues, email me ntexito@assign.org.
Sorry.
Okay.
Thanks, Nick. Appreciate your help.
Thank you very much. All right. You have to actually end it, Alicia, because I ended up making you the host. So.
Got it.
All right.
All right. See you guys.
Okay. Thanks, Tim.
Yep. Bye-bye.
Bye.